So it gives me great pleasure to welcome you to this week's Oxford Centre for Tropical Forest seminar. And our guest speaker is Katerina from the Czech Republic, from the Czech Academy of Sciences. I don't know the full affiliation now. It was the Entomology Institute of Biology at the Centre of the Czech Academy of Sciences. And Katerina did her PhD in ornithology at the University of South Bohemia for work in Papua New Guinea. Then she did a postdoc with Roger Kitching in the University of Brisbane in Australia, and then returned to the Czech Republic. And she's a holder of a prestigious ERC starting grant on the topic that you'll hear about today. So thank you for joining us, Katerina. Over to you.
Thank you very much for invitation and welcome everyone. And I will share the screen now. Should work like this. Is that okay?
That's good.
Okay, wonderful. So hello everyone. As it was announced today, I will speak about the trophic cascades in the forests. And my questions are whether the predators protect the forest canopies. Despite it's autumn now and all the canopies are yellowish, we might recall that our world is actually green. And this might be thanks to the bottom-up control, which is actually the perspective that emphasizes the disgustness and the protection of the plants and the amount of biomass in the plants protecting themselves effectively. But the other perspective might be the top-down control, which emphasizes that there are the predators who feed on the herbivorous insect. Thus, they are allowing the plants to flourish. This hypothesis is a little bit older, published in 1960s only, and it has been very controversial at the beginning, but it resulted into the good work and lots of publications on this topic. And now we can see that the truth might be somewhere in the middle. And we have actually various habitats and various types of the green, shades of the green in our world, which are shaped by the bottom-up and top-down control at the same time. So over the evolutionary race between the arthropods and plants, the plants developed various types of defenses. Some of them are constitutive, and these defenses are always present in the trees. Thus, they are very useful for the situations when the arthropods are abundant and they attack the plants very often. Otherwise, we have also the protection which is induced, and this is started in the plants only after the plant is attacked by herbivorous arthropods. And this protection is useful for these situations when the attacks are not expected in time or they are very rare. When we are looking deeper into the induced defenses, we might split them into the direct ones and indirect ones. The direct ones are aimed against the arthropods directly, killing them or making the plants disgusting so the arthropods leave. And the indirect defenses actually work through the predators. The herbivores feeding on the plants are starting the chemical pathways in the plants, releasing the chemical and maybe also visual signals to the predators or parasitoids. And these they feed on the arthropods, helping thus the plant to survive. We might therefore expect a trade-off between the direct and indirect defenses. You can imagine that if the plant itself is very well protected by the constitutive defenses, there is only few arthropods surviving, feeding on them, and thus the plant itself can be producing only very weak signal to the predators. But when the defenses of the trees itself are very weak, then there is many arthropods feeding on them. And this tree thus can signal very strongly to the predators. Thus, there might be the trees or plant species dependent more on the predators or more on the constitutive defenses. But when we are talking about the top-down control and the predators, who are actually the most important predators? We might think that the big animals like birds, maybe insectivorous birds, are eating a lot. Or we might think along the way of the biomass, their global biomass. And in that way, many people believe that the ants are the most effective predators in many of the forest ecosystems. There are also studies showing that the spider should not be neglected when we are considering who runs the trophic cascades. One study was showing that maybe the spiders and birds are actually consuming the same amount of insect per year, somewhere between 400 to 800 million tons of insect annually. And when we are looking into which predators are important, we might equally ask, where do they eat the most? And when do the cascades from the predators through the arthropods cascade all the way down to the plants? The traditional view is that the terrestrial trophic cascades are the strongest in tropics or at the low elevations where the productivity is the highest. And Oxanen's hypothesis was specifically saying that the top-down control varies as the function of productivity. This is quite an old view, but the coming reviews, which were published much later, were showing that this might not be the truth. The trophic cascades were recorded to be the strongest at the extremes and the most productive and the least productive habitats, but also only in the temperate regions. This might be simply because the predation is very difficult to study, and it's very difficult to study the whole trophic cascades. So when we start with the predation as such, we have several ways how to study that. The predation, unfortunately, is the process which is very fast. It usually happens above your heads, somewhere in the foliage where you can't see the predator and you can't see what the predator ate. So there are the indirect ways how we can expose some baits on which we document whether the predator took it or not. This might be a fly larvae or some tuna baits or something which attracts the predator, and we measure whether the bait was gone or not. Another method is the method with the plasticine caterpillars. This has slight advantage because you can recall from the bites on the caterpillar who was the predator, whether it was arthropod or whether it was bird or lizard. But unfortunately, the caterpillars are not very similar to the real prey. They don't smell exactly the same way, they look different. So another way how to study the predation and effect of predators might be the exclusion of predators. This means that you build the cages or some other barriers in the forest, and you prevent the access of the predators to the study system. And later on, you count what is the difference between the herbivory or between the communities of the insect left on the foliage. So let's start with the first method. There I was very lucky to be a part of the global study where my colleagues exposed the caterpillars in various habitats across the globe. I was surveying for them study site in Czech Republic, Papua, and in Australia. And there we observed that most of the plasticine caterpillars were attacked in the tropics and at the lowest elevations. And most of the attacks were caused by the arthropod predators like ants. This study didn't find any effect of latitudinal or altitudinal effect in the predation by birds. And generally the predation by birds on the plasticine caterpillars was very low. I always believed it might be because the plasticine caterpillars were presented very low, only 1 meter above ground. And I was wondering whether these results from the plasticine caterpillars actually match the results from the exclusion experiments. And therefore I started to survey the various predator exclosures. I found 475 experiments so far. There are still new appearing. And I focused on the studies where ants were excluded by some sticky barriers or some fine netting, and the vertebrates or birds and bats individually were excluded by cages. And when we are, these are my results, which I found from all these studies. And I found that when the vertebrate predators, birds and bats together, were excluded, the arthropod abundances always increased significantly. This is here. But when the ants were excluded, the effect was not significant. The line was touching the no effect line, and thus we didn't detect in this review any effect of ants. But when I split the data into the effect of the predators on predatory and herbivore arthropods, I saw quite an interesting thing. It looks that the birds and vertebrate predators were feeding equally on the predatory and herbivore arthropods, while the ants seem to be affecting effectively only the herbivore arthropods. And this actually, to my surprise, translated into the lower trophic level to the plant image. So the ants feeding mostly on the herbivore arthropods, then it led to the higher herbivores image when the ants were missing. This was not happening with the exclusion of vertebrate predators, where the vertebrate predators were feeding on both gills equally. Thus the trophic cascades kind of deleted each other and didn't happen and was not detected. Along the latitudinal gradient and elevational gradient, we didn't find any strong effect. There might be little one in the exclusion of ants, but nothing strong in the abundances of arthropods. But this is the picture for herbivory, where the effect was quite detectable, visible here in the ants exclosures. But doing this review, I found that the exclusion experiments which are existing now are not very comparable. They differ a lot in the methods used, and they significantly differ in the scale and in the length of the experiment. And we found that the length of the experiment specifically is having significant effect on the results. And because not many people is doing these exclosures repeatedly on several sites, I identified that this is a real gap in what we are missing to answer the questions. So I first decided to do my first experiment along the elevational gradient. And I decided to do it in Papua New Guinea, where I was previously working for my PhD on the bird communities. And I worked along my favorite elevational gradient of Mount Wilhelm, which is mountain, the highest mountain of the country, reaching up to 4,509 meters, but having the tree line at 3,700 meters above sea level. Along this gradient, we already set 8 research stations, small camps, where we were surveying the birds, and they are 500 meters elevation apart. And this is how the elevational gradient looks like from the middle. There is a tree line at 3,700 meters. We are standing with my assistant at 2,700 meters. And there is about 15 kilometers down to the foothill of this range. Along the elevational gradient of Mount Wilhelm, the habitat differed significantly. There is a very high canopy forest with very dense dark understory. And at the tree line, the trees are quite scattered with the open canopy and very dense understory. Also, the temperature decreases significantly from the mean 25 degrees per day in the lowlands to about 7 degrees per day at the highest elevational study site, which results into very different communities of predators. For example, the ants are not present above 2,700 meters above sea level. And as I, at the time, enjoyed the work with the plasticine a lot, I first started again with the experiment with the Plasticine Caterpillars. This time I was actually asking slightly different question, whether the herbivory manually made herbivory and the plants attract the predators. But here we can see also the trend, how the caterpillars were attractive and attacked by the different predators. So the attacks of the ants decreased with the increasing elevation, while the attacks by birds were kind of low plateau or quite high in the lowlands, but then decreasing also steeply, but not so steeply as ants. And there was actually more attacks by birds in the mid elevations and higher elevations than the attacks of ants. And I felt this is okay because it corresponded well with the abundances of both predators. But I wanted to do what happens if I remove these predators from the study system. Oh, pardon. So I started to do my treatments. I have got the saplings, which were control ones, to which I didn't do anything. Then the saplings from which I excluded ants by the sticky barriers. And then the vertebrate treatment, where I excluded both birds and bats together by cages placed around the saplings permanently for 6 months. And then I have got a combined treatment where I was excluding by the cages the vertebrates, and I covered everything in glue to prevent the access of the ants also. And finally, I have got those 2 tricky treatments where I wanted to remove the birds and bats separately, because this is not what has been done many times. It's quite challenging because we had to have the cages, which we were pulling up every day in the morning and in the afternoon to allow the specific predators in or keep them out. So we were hanging the cages to the canopy, we were shooting the ropes up there and then pulling the cages up several times a day. Actually, my assistants were doing that because I have got my first baby at the time, so I was navigating them how to do it. And this is kind of how the tangle glue was applied on the trunks. And my assistants distributed many kilometers of the water piping along the elevational gradient and many kilometers of fishing nets to prevent the access of predators. And as I already mentioned, the experiment was 6 and 6 months long. So after the 6 months, after the end of the wet season, we for the first time sprayed the crowns of the saplings with the Mortain fast knockdown insecticide and collected all the arthropods which we could see. And we also collected 30 leaves per sapling. Later on, we collected, like the 6 months later, we collected all the leaves and whole sapling, which we actually cut down and took the whole foliage down. The herbivory analysis is actually a work which surprised me a lot, because to measure the herbivory correctly, we needed to number the leaves and survey how the herbivory changed. So we had photos of the leaves at the beginning, then measuring the holes in the ImageJ and the Photoshop, and then we scanned the leaves and compare them from the herbivory at the beginning. And we have got many leaves to do. And at the same time, we also kept surveying the predators, what they eat, what the communities look like, and how many insectivores is actually living there. And when we get to the results, these are the graphs I plotted recently. Here, the elevation is treated as the continuous variable, and the model is kind of fitting the lines, how the effect of the predators look like. So you can see at the bottom the black line, which is showing the control saplings. Then actually not significantly different from it is the ant treatment. And then significantly different from both these treatments is the effect of all predators excluded and the vertebrates excluded. The effect was overall always lower in the dry season. You can see that the dots are a little bit less scattered and less variable because there was simply lower abundance of the arthropods altogether during the dry season. And this is a total abundance of the arthropods. When we are looking a little bit deeper into which predators were actually affected, we can see the different orders of the arthropods. And starting from my favorite one, the spiders. Spiders were affected significantly by all predators. This means that when the predators were gone, the abundances of spiders increased significantly. they seem to be fitting into the 3 niches which we made. But also there is a significant difference between the treatment of where the ants were excluded and where the only vertebrates were excluded, which means that if the ants were missing either in all or ant treatment, there was more spiders suddenly. You can see similarly that other orders affected by birds mainly were beetles. Diptera were not affected by anything. Similarly, the Hemiptera, Hymenoptera were actually kind of decreasing their abundances when the predators were gone, especially when the ants were gone. And the Lepidoptera were again rather increasing their abundances significantly when the vertebrates were gone, but also a little bit nearly significantly when all predators and ants were gone. Again, similarly, the effect on all other predators which I lumped together. So I quite like this result. It's showing me in more detail what's actually happening. And you can see on the scale here that the increase of the arthropods actually was usually like 2 to 3 times. Here the -2 means that the abundances increased 5 times. So quite a big increase. But I also like that this result is very much in line with what we found earlier from the diet. This graph is actually quite messy, but I would like to show only the yellow color standing out, which are the spiders consumed by birds. There we were sequencing the vomits of the birds which we were collecting, as well as visually identifying, and by both methods we identified big proportion of the spiders in the bird's diet, and the other taxa were a little bit less abundant. Many birds were, of course, fully frugivorous. So the spiders represent in total about 18% of the bird's intake, and the herbivores versus predators roughly 50%. And when we are looking, because I really started to like the spiders, I was also plotting how the proportion of spiders themselves changed in the relation to the other insects in the sample. So this graph is actually showing the mean proportion of the spiders in the sample. The gray area is how the control looked like, and the lines are for the individual treatments. So where the vertebrates were missing, not too much was actually happening, exactly as we have seen on the earlier graph, but the abundances of the spiders increased significantly. Again, the same results as before, just seen different way when the ants were gone. So there seems to be a competition between the spiders and ants. So when we exclude the ants, the spiders very quickly, but maybe not so quickly, are getting in and taking the free niche. The other aspect of the arthropod communities affected by our treatments was the body size. Bigger insects were surviving where the arthropods were missing. These are the similar graphs, again, the same treatments where the vertebrate cages were present by the pink and the yellow. The mean body size of the arthropods surviving were bigger. And on this graph, it's slightly different. You can see that when the birds had access to the branches, then only the bigger arthropods were surveying on this end of the graph. Similarly, my student is analyzing similar data set from also Papua New Guinea, but from different study sites, from fragments, primary forest at the higher elevation, and secondary forest. And similarly, the same way at each of these study sites, the chewers and the arthropods which have no relationship to herbivory, those which are the most eaten, are increasing their body sizes significantly in the predator exclosures. I should not forget that we were also doing those tricky treatments. And here I have to admit that we failed a bit. We were unfortunately not able to do the treatments at each of the study sites because there are no people living and the assistants didn't want to stay there for so long and works every day for me. So we had to do it at the study sites where villages were nearby. So we ended up with 4 elevational study sites only. And the results are quite messy. This is a very rough graph where the elevation is treated as a factor, so it might not be definite. But we can definitely say that against the expectation, the effect of birds and bats is not additive. The abundances of the insects increase at some of the study sites significantly, but sometimes it's because the bats seem to be most important predators at the site, and sometimes the birds seem to be eating the most. So my student Elisevot is still working and trying to match the diet of the bats and diet of the birds specifically with these results at these 4 study sites. But again, definitely not additive effect. Seems that these 2 types of predators are sharing lots of prey items together. And we are finally getting to the herbivory, to the lower trophic cascade. The graph is not showing too much because the lines are overlapping, but again, there is no significant effect, difference between the treatments by ants in comparison to control. But when the predators, vertebrate predators and all predators are missing, the herbivory increases significantly. And it increases actually usually by 40 to 70%. And the total herbivory is typically around 3 to 4% at the lowest elevational study sites and about 2.5% at the highest elevational study site. And it increases more at the elevations, lower elevations. Again, the overall herbivory was slightly lower at the dry season than in the wet season. These results are quite in line what was expected and what is known about the levels of the herbivory. The previous studies by Anstad, Schlinkert, and Kozlov found that the herbivory ranges between 3 or 1% in some specific habitats, but the Kozlov did the global study where he found this mean herbivory to be 7.6% and the highest herbivory in the temperate and lower herbivory in the tropical study sites. So this matches our results. But I was thinking whether this level of herbivory is actually significant for the plants. And there have been studies which are showing that the 5% herbivory is actually transferring into the decrease of the productivity of the plants by up to 25%. Schlinkert also observed that the 1% herbivory change translates into 10% lower abundance of the flowers in the future years. So despite the change from the 2.5 to maybe 4% is not significant, like significantly, it might be still ecologically important for the plants because every bite counts. And as I was saying, there is a very close interplay between the top-down and bottom-up control. So I would like to bring you now to the case of the study of Ficus alliana, which is the ficus which is the most abundant and most abundant ficus along the elevational gradient and spanning the longest range from 200 meters above to 2,700 meters above sea level. In the first graph, you can see how the herbivory looks like on the Ficus alliana. Normally, these are the control saplings. The herbivory decreases with the increasing elevation. Another student, Anya, was doing the study where she was trying to induce the Ficus alliana, and she was more successful in the lowlands than at the higher elevations. So here we see that maybe the plant species specifically can be dependent more on the induced defenses in lowlands than in the highlands. This goes well in line with another study which we conducted with Martin Wolf, And he found that the constitutive defenses of this specific plant species is actually higher in the higher elevations. So altogether, considering that the plant, the Ficus alliana, can be defended more into inducible defenses in the lowlands and the constitutive defenses in the highlands, we would expect the bigger difference between the control and exclusion treatments in the lowlands. And that's what we did, and this is what we exactly observed. Despite the absolute change is not a big, very big, the difference between the control and exclusion is really way bigger in the lowlands, which means that the plant species is dependent more on the help of predators is in lowlands than at the higher elevation. So then we continued like that with the other plant species, and we found that in most cases, the plant had stronger constitutive defenses where the biomass was expensive at the cooler parts of the gradient, but they relied more on the defenses through the induced defenses in the lowlands. So we can conclude that the predation by vertebrates decreases with the increasing elevation. We don't see big effect of ants, but that's because they are eating only herbivores. And there are the spiders who are fitting in and taking over. Thus, the overall effect of the ants might not be detectable. So overall, the predation by the herbivore of ants is actually bigger than the effect of the vertebrate predators on the herbivory sometimes. And so the birds, again, vertebrate predators are feeding on both predators, herbivores at the same time, keeping the ratios in balance. So there is a very complex interplay between the top-down and bottom-up control, which works on the basis of individual plant species. And the overall cascades, whether the cascade will be there or not, depends on the composition of the plant species which we select for the exclusion experiment. And really the vertebrates, especially birds, seems to be the most effective predators. As for the ants, both from the review and from our experiments, we found their effect is dependent on the length of the experiment. The longer the experiment was, surprisingly, the smaller the effect was, which means that the spiders obviously were taking some time to fit in and decrease the effect of the ants by themselves, while when the experiment was very short, up to 1 month, we typically found a very significant effect of the ants exclusion. But this is what I was doing so far in the forest understories, and what really made me kind of angry and curious is that most of the predators and most of the trophic cascades are actually expected in the forest canopies, and I could not reach them because I had no way how to get there. It would be very complicated, and that's why I designed the next experiment where I started to use the canopy cranes, and I tried to replicate the same experiments at several canopy crane sites. I was doing exactly the same treatments, again aiming to exclude birds and bats separately at some strata, 6 treatments in total. And there is actually quite a handy network of the cranes, and I was so far finishing my experiment in Tomakomai in Japan. I finished partly in Bupeng in China, but from there we didn't manage to export the insects, so we have only the final results on the herbivory. We are still working in Kakoba crane in Papua New Guinea, and we finished successfully full 1-year-long experiment at both Australian cranes in Drain Tree Research Observatory, Rainforest Observatory, and at Uke Face in Sydney. Because the Lambert Hills crane was closed in the meantime, and we still wanted to continue our research, but could not go to the tropical sites anymore, we decided to replace it by the Leipzig canopy crane because it has the similar plant communities as the Tomakomai, and is accessible to us during these difficult and challenging COVID times. So this project is still under the way. I will show you only the partial results. But to start with, we needed to standardize the plants phylogenetically. So we selected from different families specific plant species on which we work. And then we started to exclude the predators from them. We needed to consider also their abundances because we need 70 individuals of them and at least 12 individuals of them within the arm of the crane, which is sometimes a big challenge. And so far, as I said, the project is still ongoing. We work with 720 saplings at each of the study sites and about 350 branches at the Canopy. And we so far used 55,000 kilometers of PVC pipes, which we shipped to the sites from either China or from Czech Republic. 200 kilo of tangle glue, which we glued on the trees. And we have got several nice hundreds of thousands of insects, which we continue to identify. I'm actually pretty fast, so still enough time there. So I would like to mention that we started the experiment again with the work on the Plasticine Caterpillars. I just like it. And I was always again wondering whether it works or not. So at each of these study sites, we exposed 6,000 caterpillars in the forest understory and forest canopy, and we observed who predated it. Here is the total predation, and you can see very interesting results, which I totally didn't expect, but we detected the higher predation in canopy only at the sites which are temperate or like nearly temperate in Tomakomai and Yukphase. And actually I have here old picture. We have also now new results from last month from Leipzig, and there the results are exactly the same as in Tomakomai. So 3 study sites where we have higher herbivore predation on caterpillars in the canopy. And then we have got the 3 tropical study sites where the predation by on plasticine caterpillars was actually higher in the understory. I didn't expect that, I have to say, but I will keep the reasons and the explanations for the publication, which should be coming out soon. But it correlates well with the abundances of the arthropods and the predators which we are observing there. And as we knew a bit about what kind of predators we are dealing with, what we could expect on individual plant species, what are their defenses, we started again to exclude the predators. As you might expect from the predation on the plasticine caterpillars, we had to kind of zoom in when we were looking at the results from the understory. In the understory, the overall predation and the effect of predators was very low in the understory. But when we zoom in, we can see the control. Nothing was done to the saplings. No, like roughly 10 individuals of the arthropods on meter square of the foliage. But when we excluded the predators, ants, again, nothing happened as in our previous experiments. And then when the birds and bats were excluded, the abundances actually increased significantly, but the highest abundances we observed when both of these predators were absent. What was happening in the forest canopy was rather striking, not that we increased the abundances by 5 individuals per meter square, but by actually 140 individuals of the insects per meter square. Especially when all the predators were gone from the forest canopy, the jump was significant. But again, despite there was many ants in the canopy, they didn't do anything with the arthropod communities. Here, my student prepared me today a very basic graph, which is actually showing some details of what is happening in there. We found quite interesting difference between which groups of the arthropods are where. We actually found many more beetles in the canopy than in the understory, but more caterpillars, Lepidoptera larvae feeding on the understory than in the canopy. So overall, there was more caterpillars than beetles, and there were mostly caterpillars in the forest understory. So if you can imagine it like this, many caterpillars here in the understory, only few of them up there in the canopy, quite many beetles in the canopy, but only few in the understory. But when we are considering what kind of damage they could cause to the plants, we have to remember not all the beetles are actually herbivores. We still will have to do some finer work and identify to which guild they are belonging. But what do you think? Where there is more birds? Is there more birds in the canopy or in the understory? I will reveal it now to you. There is actually 30 kilo of insectivorous birds in the forest canopy versus 5 kilo per the same area of the insectivorous birds per in the understory, which goes pretty well again together. There is more predators, thus there is very few insects living in the understory, and the caterpillars are kind of hiding in the understory and surviving and avoiding the predation pressure, which is way higher in the canopy. This has the direct consequences for the herbivory, of course. And this is actually the last slide, which I would like to show you today. This is the herbivory from Tomakomai, and it's quite messy, but you can see here the treatments, individual plant species which we were working with, and then the canopy and understory. The biggest variability of the herbivorous damage was explained by the plant species, which means by the bottom-up control. Whether the plant is very yummy or very poisonous to arthropods is determining how big herbivory it will be suffering. So there is Ostraya japonica, which is eaten well, but there is a magnolia, which is not eaten at all. After that, the second variable explaining the variability is the strata. The canopy trees of the same species are always eaten less than the saplings in the understory. Yeah, understory herbivory is higher than in the canopy, of course. And only after that, the effect of the predators could be detectable as the third important factor. Here it's again quite detectable difference between, like no detectable difference between the actions of the predators and versus control, but then the herbivory increases if the vertebrate predators and all predators are missing from the study system. So similarly as we were seeing before in the previous experiments, there is a very tight interplay between the bottom-up and top-down control, and it very much depends on the natural abundances of the arthropods and on the abundances of the predators, which we can detect at each of the study sites. We will continue the work on the more tropical study site in Australia, where we are finishing soon. So if you would like to follow our research, we will be very happy to hear from you. And I hope that I showed you today that these trophic cascades are quite complex, could be detectable and dependent on one plant species, but cannot be sometimes cannot be detected as in the whole population, because the effect is hidden based on what kind of plant species is presented in the experiment. So quite difficult to detect them. And with this, I would like to acknowledge all assistants who helped us in the field, who finished the experiments for us in Australia, especially when we were not able to get there on time due to COVID, and they send us all the samples. And also to all my students who work hard on identifications of the arthropods which we are collecting, and also on the analysis of the herbivory, which is a tedious work. And thank you very much for your attention.
Okay, thank you very much. Thank you. Absolutely fascinating. Really, really interesting. Let's stop sharing the screen. Great.
Yes.
And I'll invite everyone to ask questions, and also do switch on your camera if you're comfortable with that. It's quite nice to have a visible audience for the Q&A session. And we can ask questions there. Great, but thank you. Really, really clear, and lots of really fascinating new insights, and quite a few surprises there as well, I thought.
Yeah, also for me.
Yeah. So I'll open the floor to questions. If anybody has any. While we're waiting, I'll kick off with one. I think it was the end. I think it was from Tomokomayo. You were showing a result where in the canopy, when you excluded all predators, you had a really big effect on arthropods. But when you just excluded birds, it was quite a modest effect.
Yeah.
If I remember. Does that mean it's the bats that are really affecting that process, or am I misunderstanding?
There is actually many bats, but that was one of those surprising results, because the ants by themselves didn't have any effect, and then when I excluded ants and the vertebrates together, the effect kind of increased in comparison to the vertebrates. So there might be actually also some methodological issues because there was many apids, and they seem to be playing a lot with the data, but simply another thing is that in the cages where the ants were missing, there was many Hymenoptera larvae at that time, which were just hatching from the eggs. So there were branches where there was hundreds of them, and they drove the results too much. So I think maybe because the birds are not effective in eating the small eggs, but ants are. The ants, just because of the timing, because the ants were not there, the eggs hatched, and there were just these tiny things which were still not eaten by the birds, would not be eaten by the birds at the time, but were caused by the absence of the ants.
Okay.
Yeah, sometimes I feel that abundances of the arthropods results are very dependent on what time exactly we go there. Not only we of course aim for the nice weather, but we can go 1 week later, 1 week earlier, and we catch completely different phenology. That's why we try to replicate this experiment several times, several seasons, to get the proper picture.
And then any one sampling time, how long was your period? Was it a few weeks or 1 week?
No, this was like in the temperate study sites is the full season. Okay, and good. in the tropics, it's 6 months, like full year.
It's quite long periods. Yeah.
It's a long for Tomokomayo. It's quite short because the leaves appear the last week of April, and they are actually dropping in the late August. So there is a few months, but we can't go further because then we would not find the leaves with the numbers anymore. So we do the max. And then other study sites, we try to match it more or less like 5-6 months.
Mm-hmm. Great. Okay, anybody else got a question?
I have seen here one in the chat.
Oh, yes. So Shyam, do you want to ask your question directly?
Yeah. How many leaf samples per plant are you taking? Because I also work with Herbivory with my Brazilian colleague, and I also working with Dr. Will from US, this Herbivory variability work. So how many leaf you are leaf sample per plant is calculate using for the estimating the Herbivory?
We do 30 leaves per sapling or per branch.
Okay, and there is some kind of, you are collecting randomly, or there is just some kind of the protocol?
No, there is so we pick randomly a selected branch, which of course, like we just look at it and pick the one which very likely will not be disturbed by the treatment. Mm-hmm. And from that branch we select 3 like that. And from each of these 3, we mark first 10 leaves from the top, and then we tie a string. Yeah. So because sometimes the permanent marker gets washed, So we know where the 10 leaves finished. And after that, we calculate the herbivory on these 10 numbered leaves, and we calculate also those leaves which grew above the leaf where we started.
Yeah. Thank you.
Yes. So it's like 30 leaves per sapling by 760 saplings in the understory. Everything 6 times it accumulates.
Yeah, we recently have collected this tropical tree herbivory, that paper published in ecology. So the data paper we published, and maybe we will calculate. So it is all about. So we took a lowest branch from the distance we marked, and then we calculate the herbivory from bottom. Take a branch from bottom 1, 2, 3, 4, 5, 6 like that, and take up 50 leaves. If there is no branch, no 50 leaf in one branch, then we take a neighborhood branch, and so to reduce the biasness for the herbivory.
Exactly. In the previous study, which I was showing by Markéta Tahadlová, there was 50 leaves, but simply because we never fit 50 leaves into one small branch, and it will be nearly double of the amount which we have, which is obviously not feasible for us nearly to analyze. Then we had to stick it to 30, which is still representative based on our previous results. So
are you using some kind of the app or the image?
No, the app is unfortunately not useful for us because we do it quite in detail at some of the study sites. We try to distinguish the herbivory by minors and by chewers. So we are using manual work, many students laboring in Photoshop and an ImageJ.
Yeah, I also work on ImageJ to be much easier.
Yeah.
Just to follow up on that, I was going to ask a question about that. So you're scanning the leaves for lost leaf area, and then for the minors and gall feeders, is it by area you're quantifying, or just the number of counts that you have of those?
No, we kind of mark the area which is visibly, either it's a gall, which usually doesn't, sometimes it makes like that yellowish patch around. So that's what we mark as the herbivore's damage, but of course it's sucking through the leaf, so it can be quantified. But because we have got another student who is specifically focused on the galls, then we have got also the galls and the diversity of the galls and the abundances per leaf, abundances per branch. So this all could be added to the herbivore's damage. Yeah.
Okay, that's it. And so I guess one part that gets missed is sap feeding, I suppose, and those components. What fraction of the total herbivory do you think you capture with your methods? And are there some elements that are inevitably missed because they're harder to quantify?
No, actually, I don't know. That's a very good question. How much we are underestimating? Yeah, definitely. And unfortunately, it will be very different from site to site because there was many more aphids in Tomakomai than elsewhere, and there was many more galls in Sydney than anywhere else. So these are total two extremes, which we could record where the herbivory, we are obviously missing a lot.
Right. Interesting. Okay, Owen, question.
Hi, thanks, Katerina. Really, really nice talk. Amazing data sets, especially when you have to measure all of the herbivory on marked leaves. So I have one question related to that, and then if I can ask a cheeky second question as well. So is there ever a case for assessing herbivory using standard standing herbivory, so just doing like a snapshot survey? Or is that, because that's obviously a lot quicker and easier,
yes,
is that always going to be problematic, do you think?
Before I was using that, but in the earlier experiment, when I didn't mark the leaves yet, it actually happened to me that, you know, I collected the leaves at the beginning, then I collected other leaves at the end, and the herbivory was totally off, like very different. So we decided that to get the exact number, we really need to follow the leaf. Also, because the leaves are dropping, and sometimes the whole leaf disappears and we don't know, but from the branch when the leaves are marked, we can kind of estimate whether the stalk is still there, whether it was completely eaten or not. So it's quite tricky. Yeah, I would like, from a few years ago, I started to do only the rate of the herbivory, but I understand that the standing herbivory is much easier.
Okay, thank you. And then my follow-up, so you talked a little bit about potentially missing some herbivore categories like the sapsuckers, and presumably below-ground herbivory also could be a significant factor. On the predation side, or the natural enemies, I guess parasitoids are an obvious category of natural enemy that might induce top-down trophic cascades. Do you have any idea whether they're showing congruent patterns in terms of their impact to the insect predators and the vertebrate predators with elevation, latitude, stratum?
This is very good question, and there is a symphony who is specifically working on this for me, but because the work is very slow, he has no results yet. But all the caterpillars which we are collecting, he is from Tomakomai, he dissected them and searched for the parasitoids. The other sites are being sequenced, and he is searching for the pattern in there. And so we have the caterpillars for treatments and caterpillars from the control branches, hoping to see if there is some difference, whether the caterpillars have bigger maybe chances to survive somewhere, but so far he's in the way like sending the caterpillars parasitoids to the bolt. No, I can't say more about this currently. I hope he will get faster.
Great, thank you.
That's incredibly painstaking work. Okay, there's a question from Charlie that I'll read out. Just to clarify, you believe that canopy arthropod abundances are lower than understory, primarily due to predation pressure. Did you take any measurements for microclimatic metrics, as this would also seem to be important, particularly in the tropics?
Yeah, I actually immediately recalled it simply, it doesn't have to be because of the predation, of course. There are many things which are making the canopies very disgusting for the arthropods to live. There is a strong wind, there is lots of radiation, and as we are working on the edges of the canopies, we can't get with the cages into the canopy, of course, and also the crane can't get us in. So we might be missing some part of the variable there because of the wind and of this radiation. The arthropods might be living a little bit more inside in the canopy while we are working outside at the edges. So, but outside on the canopy, it might be exactly because of the climatic variables also, and we are getting them in the standardized way from the cranes. All of them are measuring somehow the wind speed, the radiation, temperature every 5 meters. So this is what we will be incorporating later on. Also, one thing I completely forgot to mention, we are collecting all the leaves, which we are working with at the end. We, together with Euhapeca salminen, we are analyzing the polyphenols, alkaloids, and oxidative activity of these leaves, both in understory and in the canopy. And the students are measuring the length and density of the trichomes and CN ratio. So, these factors will be incorporated in, and hopefully they will explain the significant part of where the arthropods are and where they are not.
Yeah. I imagine, as Charlie suggests, the radiation pressure is stronger in the tropics, and the wind, I mean, maybe stronger in temperate. Do you see stronger stratum contrast in the tropics or in the temperate sites? I can't remember what you showed.
Very strong in the temperate.
Okay, right.
Yes.
So,
no very strong. Generally similar responses in the canopy, understory, and tropics, but very big differences in the temperate.
okay, so there's a stronger temperate effect. That would suggest radiation isn't probably the main factor. It could be wind or other things. Okay, So a couple more questions, Kate.
Hi, hi Katerina, that was brilliant.
Hi.
Thank you.
Thank you.
Really, really nice to hear about it. And I totally appreciate the amount of work involved in all of that. So you've got these lovely canopy sites and you showed us the Japanese data, which is kind of more, it's a temperate site,
Yes,
not
yes.
tropical. So what are you expecting in terms of a tropical temperate contrast overall? Predicting and why?
What I see is that the changes in the abundances of arthropods naturally are not so big between the canopy and understory in the tropics. But there is way more insectivorous birds living in the forest understories, which might be causing some effect. At the same time, there is many more ants in the tropical canopies than in the temperate canopies.
Yeah.
So either there will be very tight interplay where these types of the predators will somehow share, and the overall effect will be nothing, simply the birds taking more in the understory and ants more in the canopy. Or yeah, I actually do not expect big vertical trend in the tropics. Maybe.
Do you expect,
Yeah.
so yeah, you're sort of expecting it to be different than to what you've got here, really quite different.
Yes, quite different from, that's what I can already see. It's quite different for many taxa, actually opposite to what we have seen in Tomakomai.
Yeah, and then when we suppressed our ants in the canopy, we got really big increases in the invertebrates in the trees.
Yes, I feel the same.
Yeah.
I exactly like this. This is what seems to be visible there, despite we didn't run the analysis on it yet.
Brilliant, thank you.
Did I miss it? Why do you think there's more? Well, why is there more bird predation in the understory in the tropics?
This is simply a physiological thing because the insectivorous birds in the tropics might be more adapted to the forest interior. And many of these insectivores are really small birds. Like if I recall this Papuan side, there is many of these sericornices, flycatchers, which are in the forest understory. Those birds which are in the canopy are mostly bigger. Partly frugivorous, going after the fruits, nectars, and taking the insect only occasionally as a part of the diet when feeding the chicks. So overall, I feel the bigger pressure in understory in tropics.
Okay. And Vivek?
Yeah. Thank you very much. Thank you, Katerina, for this insightful presentation. I'm a researcher working in MS Swaminathan Research Foundation in India. I work among small farmers. So we have research sites across the altitudinal gradients also. I was just wondering, is it too early, or are there any insights that agriculture systems could gather from your study about forests? Related to herbivory and predator and these kind of gills. So that is my first question. And my second question is, are there any citizen science or community science tools being developed to assess the process like predation, so that there is possibility for many people to participate from underdeveloped and developing countries also through apps like iNaturalist? Yeah, thank you.
That is actually a pretty cool question, the second one. Unfortunately, I don't think that's something, it's very sad to say, but nothing useful could actually come from my study, because the diversity is so big, and despite we have got tens, hundreds of thousands of insects, there is nothing like detectable outbreak or like really abundant thing which could be considered as being important for the pest or like agricultural work. Because also we work in we try to work in the very natural, not agricultural habitats. And as for the questions, we are actually dreaming about that. We don't have the app, but one of my later, a little bit older PhD student is actually thinking to expand a similar project with Bastien Castagnero, who has got this predation project across the whole Europe on the oaks. And they are thinking this is a very nice citizen project, but unfortunately only in Europe and focusing on predation. So they try to get wider. Hopefully it will get through some funding, but so far no app available.
Yeah, thank you very much.
Okay.
Yeah, thank you.
Elena?
Elena? Oh, yeah.
Okay, thanks
Hi,
for the really nice talk. I want to change a bit thing. So I was interested in the spiders that you mentioned in the beginning of your talk.
Mm-hmm.
So because I've been in Papua New Guinea and I've seen that there's all kinds of spiders. So do you know anything about the spiders that the birds like? So do they eat more the small spiders or bigger spiders or poisonous spiders or non-poisonous spiders? Or
They eat relatively small spiders, around 7 millimeters big spiders. We didn't detect any big spiders. They mostly eat the spiders which are on the foliage. They get them from the gleaning, not the web making spiders too much. And yeah, the results are quite nice. I like it. I was actually a bit worried when we put the exclosures and the cages, then there will be many spiders getting in because they will use the cage construction as the place where to put the webs. But fortunately, it was not happening at all. So they were really, those spiders which are eaten a lot, as we see from the diet, were actually really increasing their abundances in the exclosures. Yeah.
Thank you.
Thanks.
And I think I must have missed this in your methods. How are you excluding ants? What's your protocol for?
That's the tangle glue, the sticky barrier, which we apply either on the trunk or on the branches.
Okay, I see.
Yeah, so unfortunately, that's also limiting any other crawling arthropods, which could just get glued there. And in the canopy, it also gets washed away. So it needs to be checked from time to time. We were also making some protective layers, how to prevent the rain. So it was a bit tricky. Yes, yeah, but that's tangle glue.
Mm-hmm. Okay, great. Any more questions? If not, I think we can draw to a close. I encourage you, in our seminar tradition, to unmute your microphone and give an audible round of applause to our speaker today. So thank you very much, Katerina. That was absolutely fascinating. There's lots to think about, and I look forward to seeing the papers and the studies emerging on these sites.
Thank you very much.
Great. So we'll record this and I'll post it onto YouTube later today. And feel free to share and circulate widely to those who couldn't be here. And hope to see some of you next week at Sandra Diaz's seminar at 4 p.m. same time next week. So thank you, Katerina. Thank you, everyone.
Thank you for invitation. Bye. And have a great weekend.
Okay. Bye-bye.
Bye.